Swayam Prabha. Digital India. Educated India.
So, today we will be talking about the role of effector molecules in the kinetics of enzyme catalyzed reactions. So far, you will recall that we have seen the role substrate molecules play to influence the rate of a enzymatic reaction. They bind with the enzyme and have dynamic complexes that ultimately break down to form the product. Now, this complex enzyme substrate complex formation can be affected by a number of other external molecules which are not really directly the players in the enzyme reactions. That means, neither they are substrate, may or may not be products, and such molecules are called effector molecules. Essentially, those molecules whose presence in the reaction mixture influences the rate of reaction. If it is increasing the rate of reaction, we call them activators. If it is decreasing the reaction rate, we call them inhibitors. Now, obviously, the role of these molecules is very important in the kinetics of enzyme catalyzed reactions for certain reasons. One is that they give you a picture on the reaction rate in the presence of those molecules. Rather, you can regulate the reaction rate to a enhanced value or to a decreased value in the presence of those molecules. In fact, in the living cells also, these molecules provide a regulatory device to control the rate of reaction. We will see when we go on subsequently how many of the molecules they control the rate of reaction by inhibiting or activating a particular enzyme in the beginning of a metabolic pathway. In addition to that, the understanding of the enzyme inhibitors or activators give us very precise information on the specificity. Most of the information on the architecture of the active sites, which are the functional groups that are present on the active sites, or the mechanism of the specificity has resulted from the understanding of the interaction of the inhibitor molecules or activator molecules in the enzyme catalyzed reactions. Particularly in the case of multi-substrate reactions, which we will be dealing subsequently, where more than one substrate molecule takes part in the enzyme catalyzed reaction, their kinetic mechanism is also understood by the role of certain external effector molecules. That is another major advantage. So, these are the issues that provide us a very important understanding towards the role of effector molecules. But even on the practical aspect, probably from the industrial aspect, also effector molecules are very important, because a large number of drugs, the preservatives that are used commercially, they basically work on the principle of modulating the enzyme reaction rate. I mean, we are aware of many of the sulfur drugs. In fact, all the sulfur drugs are basically inhibitors of one or the other enzyme operating in the living system. The only difference is, the only care that is taken in designing a drug, that the particular molecule which acts as an inhibitor for a particular toxic substance, or let us say for an infection, should not be toxic to the human beings, otherwise it cannot be used as a drug. And most of the materials that we use, whether they are sulfur drugs or antibiotics, or they are essentially inhibitors or activators of certain enzymes. So, from that point of view, even understanding their interaction is quite important. Now, if we take I mean, in the first instance, the first item, that is your activators, and now these are the molecules, as the name suggests, that enhance the rate of the enzyme catalyzed reaction. Now, some of the activator molecules may be absolutely essential for a given enzyme reaction, some may not be so. They may only provide stability to the enzyme substrate complex, and therefore, the nature of activator molecules might differ from case to case. Now, the typical examples of some of the activators are, for example, in the case of alpha amylase, calcium ions act as activators, which essentially is known to stabilize the enzyme substrate complex. In the absence of calcium ions, the enzyme substrate complex is not very stable, or the dissociation constant of enzyme substrate complex is relatively high, and therefore, the rate of reaction will be very slow. Similarly, in the case of most of the kinases, hexokinase, a typical example, the very first reaction in glycolysis, magnesium ions are absolutely required. That is another example of an absolute requirement of an activator. In the absence of magnesium ions, the enzyme will not be functionally catalytically active, and therefore, these activators, they function by participating or facilitating in the either enzyme substrate binding or in stabilizing the enzyme substrate complex.
Sir, so what is the difference between then cofactors and activators?
The cofactor, essentially the difference between cofactor and activator is, a cofactor will have to participate in the catalytic mechanism. I mean, that means for example, take for example a cofactor like NAD. Now, it is used in the oxidation reduction reactions, and in the absence of any such residue in the active site which can participate in the oxidation reduction reaction, the NAD provides a molecule which undergoes redox reaction and therefore catalyzes the. So, therefore, it also binds, it also plays a role in the substrate binding, and but as a matter of fact, the and the role of cofactor is absolutely essential. The reaction cannot take place in the absence of cofactor, where in the case of activator, we are only enhancing the rate may not come to 0, it might have a very small insignificant rate in the absence of an activator, and in the presence the rate might enhance.
So, Mg2 plus is not necessary in case of kinases.
Mg2 plus essentially, as I mentioned, is an absolute requirement. So, it falls in the category of both. Actually, it also acts, it also participates in the catalytic mechanism. So, in fact, that is an example, but other than true example of activator will be essentially calcium ions, which do not take part in the catalytic function, but provide a stability to the enzyme substrate complex. That means, binding process is facilitated. So, I think magnesium is a something on an interface. And so, now to look at activators from the mechanistic point of view, the role of activators can be considered as binding to the enzyme molecule to give you an activated enzyme. That means, the enzyme molecule, free enzyme molecule, is basically inactive or has a very low activity. When the activator molecule binds to it, the EA complex becomes an active enzyme complex. Now, then this EA complex will essentially undergo, for binding of substrate, which is the active enzyme substrate complex, and then this enzyme substrate complex can break down to the active enzyme plus product. In the classical K1, K minus 1 and K2. Now, if we consider that the dissociation constant of activator binding is Ka, the dissociation constant of activator binding to the enzyme is Ka, then in that case the rate of such a reaction you can express as Vi is equal to Vm into S upon Km 1 plus Ka upon A plus S. Now, you ultimately end up in a rate expression in which the Km term is modified. Now, this you can written as Vm into S upon K prime m plus S. So, essentially the role of an activator essentially works out to modify the Km value, intrinsic Km value of an enzyme, and obviously the effect on the Km value will influence the rate of the reaction. Now, one can also see that the modified Km value is essentially the intrinsic Km multiplied by 1 upon Ka over A. That is, when A becomes very large, the activator concentration present in the reaction is much larger than Ka in comparison to Ka. That means, when you have sufficient quantity of activator present as required, the K dash m approaches to that of the intrinsic Km, or K dash m upon Km is equal to 1. Also, when the activator is 0 or when there is no activator present, the enzyme reaction will also not take place, because the modified Km value will be too large. There will be no effective binding taking place, that means this reaction will be, the second step will be ruled out, and therefore, the activator molecules can influence the reaction rate in a process, in an enzyme catalyzed process, by this simple way to modify the Km value. The second category of effector molecules, as we noted, was inhibitors. Now, essentially, as the name suggests, these are those molecules which decrease the rate of an enzyme catalyzed reactions when present in the reaction mixture. Now, essentially they form complexes with enzyme molecule, enzyme substrate molecule, both or one of them. Now, they form dynamic complexes with the enzyme and enzyme substrate complexes, and thereby influence the rate of reaction. Typically, the inhibition patterns or inhibitors can be classified into two main classes. One are irreversible inhibitors, which essentially means that the inhibitor molecule binds to the enzyme in an irreversible fashion and cannot be recovered or removed by physical operations. Very often, when to a reaction mixture, if you have added a inhibitor molecule, one can remove it by a physical method, say for example by dialysis or ultrafiltration or any of those methods, but irreversible inhibitors cannot be removed once added to a reaction mixture, and their effect is irreversible. That means, ultimately they will tend to reduce the rate of reaction and provide a poisonous effect on the rate of reaction. In fact, sometimes they are also called catalytic poisons, because they irreversibly poison the catalyst. Of course, in most cases the effect is such that the reaction rate or the catalytic function of the enzyme approaches to inactive form, and thereby they cannot be used for catalytic functions. Now, basically the irreversible inhibition is caused by modification of chemical modification of one of the amino acid residues on the active site. Most of the chemical modification reagents that are used for modification of the enzyme active sites do play a role in the irreversible inhibition. Most of the alkylating agents, they will alkylate some of the amino acid residues and thereby make the enzyme inactive. many of the heavy metals like mercury essentially also acts by, or some of the poisonous gases, they also have the same action of irreversible inhibition, whereby I mean saving the person is difficult. Now, again on the same lines, the role of irreversible inhibitors can be expressed as E plus I going to E I, an enzyme inhibitor complex which goes ultimately to inactive enzyme. The first step is a reversible process, but the process does not stop here. Once E I is formed, it goes into a inactive, a first order irreversible step which inactivates the enzyme, and here one can write the concentration of E I at any given time can be expressed as E 0 minus E I into I upon K I, and where K I is the resolution constant for the inhibitor, and then one can write the inactivation rate as your K inactivation rate into E I, that is equal to K inactivation E naught upon, you can just work out I mean simplify this expression in for E I, and you will get 1 plus K I over I. So, therefore, if I is in excess, if the magnitude of inhibitor is in excess such that you can consider there is a constant, then the whole expression essentially is a first order rate constant, that is your K D into E naught, where K D is essentially a first order rate constant, assuming that I is in excess and can be considered as constant, and such reactions are very often noted to be poisonous in nature. The most of the substances that are technically called poisons, whether it is mercury or cyanide or nerve gas or things like that, they all behave in the form of irreversible inhibitors, and essentially from the industrial point of view, they are really accepting to understand their role to I mean evolve methods to prevent the poisoning. Otherwise, they do not have much of industrial value. It is mostly reversible inhibitors which play a key role in most of the industrial I mean processes. If you took the second category of reversible inhibitors, now here the inhibitor binds to the enzyme in a reversible manner, and when I say reversible manner, I mean that after addition of the inhibitor to an enzyme substrate mixture, one can physically remove it by physical methods, most commonly use ultrafiltration or dialysis, such that the enzyme is fully restored to its initial activity. Of course, in the presence of inhibitor the activity will be decreased, but once you remove it physically, the original activity of the enzyme is recovered. In a sense, these molecules make dynamic complexes with the enzyme which have different catalytic properties to that of the native enzyme. Now, essentially they bind in different ways, different inhibitor molecules bind differently to the enzyme molecule or enzyme substrate molecule complex, such that the catalytic properties are altered and thereby the rate of reaction is altered. Now, a very common pattern to study reversible inhibition, in fact our total orientation to study the inhibitors will be mostly directed towards understanding the reversible inhibition, because as I mentioned that it is this reversible inhibition which forms the major portion of the effector molecules that are of industrial, medical or biological value in the case of even understanding the scientific basis of enzymatic catalysis. Now, how do we study? I think there are certain general patterns which we follow in studying reversible inhibition. Before we go to the individual cases, I would like to highlight that, and the first is that I think the first thing that we must look at the pattern of binding of inhibitor to enzyme or enzyme substrate complex. That means, how does the inhibitor, a particular group of inhibitors, they bind to the either the enzyme molecule. So, that means essentially you are proposing an operational reaction mechanism which may be slightly different than that proposed by Michaelis-Menten or others earlier as we discussed, and that mechanism need to be understood very clearly, and that will give you a physical picture of the enzyme substrate binding and what happens. Then the second thing which comes now, basically on the basis of this binding process, you develop a rate expression, and obviously the rate, we always tend to compare a rate expression with that of a Michaelis-Menten rate expression and try to compare, and then look at the effect of the inhibitor concentration on Km or Vm or both of the reaction. See, first we have looked at the binding pattern. Based on the binding pattern, we develop a rate expression, compare it to the Michaelis-Menten equation, and see how does the Km and Vm of the native enzyme are influenced by the presence of inhibitors, and then we see the effect on Lineweaver-Burke plot. Again, that is another I mean sort of a basic expression of a kinetic behavior in the case of enzyme catalyzed reaction, that you consider if the rate expression that we have obtained, if a double reciprocal plot is plotted between 1 upon S and 1 upon B as a function of inhibitor concentration, how does this represent? And the fifthly and probably another important pattern will be for each class of inhibition, degree of inhibition. That means we define a term called I, which is essentially nothing else but V naught minus Vi upon V naught. If V naught is the rate of uninhibited reaction, classically Michaelis-Menten kinetics, and Vi is the rate expression for inhibited reaction, and the degree of inhibition that is produced by the presence of inhibitor is obtained. Even from a theoretical rate expression, you can work out an expression for degree of inhibition, and that will have a characteristic pattern which can distinguish a particular type of reversible inhibition. So, basically all our reversible inhibition studies will be based on these 5 approaches in general, of course. And now now I take probably one of the most simplest and the most commonly encountered pattern of reversible inhibition, what we know as competitive inhibition. Now, as the name suggests, here the competitive inhibitor combines with the free enzyme on the same site, which is what we understand as substrate binding site, and so therefore the inhibitor competes for the binding site with the substrate, and usually both inhibitor and substrate molecules are mutually exclusive and compete with each other. Now, very often we feel that the enzyme binding site, the inhibitor binding site on the enzyme molecule and the substrate binding site on the enzyme molecule are identical. Now, this should be taken in a very broad sense, in the sense they could be on the same site where the substrate binds, or even they could be on a site in proximity of that, or in such a way that the binding of the substrate is influenced. It is not always necessary that the binding site has to be exactly the same as that for substrate. The purpose is that the binding of the inhibitor, the presence of the inhibitor makes the binding of substrate difficult in a competitive mode. That means, maybe it may be in the proximity and such that it has such the inhibitor molecule has such a conformation that the original binding site is disturbed and substrate binding is not feasible. Or alternatively, after binding of the inhibitor molecule, the substrate binding site might be modified in such a way that substrate binding is not feasible. All those cases, variety of cases, will come under the category of competitive inhibition, and the purpose is that the presence of inhibitor must prohibit or reduce, not prohibit or reduce, because it is a reversible inhibition depending on the concentrations of the two species, inhibitor and substrate, they will affect the binding mechanism. A very typical example and probably the most easily understood example is of your oxidation of succinic acid to fumaric acid, that is your CS2COH, CS2COH, succinic acid, that oxidizes by a FAD catalyzed reaction to creation of a double bond plus Fa, which is a reduced FAD. And now this enzyme is succinic dehydrogenase SDH. Now this is inhibited by a very closely resembling molecule, closely resembling to the substrate, malonic acid, which is essentially CS2. It also has two carboxylic groups, but only one methyl group. Unlike succinic acid, it is very similar in structure conformation, and very often the enzyme active site mistakes it for succinic acid, and therefore it acts as a competitive inhibitor for in the same reaction. Another example of a competitive inhibitor is sulfanilamide. Now that acts as a, which is essentially a sulfur drug. Now that acts as a competitive inhibitor for folic acid biosynthesis from para-aminobenzoic acid. From para-aminobenzoic acid, now the amino group on the aromatic ring is identical in both the molecules. This is sulfanilamide, this is para-aminobenzoic acid. Now essentially this para-aminobenzoic acid is starting material substrate for synthesis of folic acid. Now this synthesis is inhibited by this enzyme and by this inhibitor, and therefore now such a synthetic reaction route although takes place in microbial systems, but does not take place in the human beings, and therefore this acts as a very potent sulfur drug. That means because this reaction is not available in the human beings, but microbial systems are totally dependent on synthesis of folic acid, it can easily inhibit the growth of microorganism, and sulfanilamide is a very potent antibacterial agent in the case of human systems. Now if you look for the reaction scheme or the binding pattern which is followed by competitive inhibitors, we can write the first stage as enzyme substrate binding goes to ES, goes to E plus P, then the same enzyme molecule, free enzyme molecule, binds to the inhibitor and gives you EI. We can just consider, let us say, dissociation of ES in the form of Km. Obviously when I say Km, I am referring to K minus 1 plus K2 upon K1. Just consider it is analogous to the dissociation constant. Just for simplicity I have written here, and this is your K2, this is your Ki, the dissociation constant for the inhibitor. Essentially you can, or rather be very clear as Ki is equal to E into I upon Ei, and Km is, or Ks in that matter, is E into S upon Es. Now, you can write the rate expression for such a binding mechanism, and the rate expression will come out to be V naught equal to or rather V naught upon Vm equal to S upon Km into 1 plus I upon Ki plus S. So, basically you notice that if you now you compare it to the Michaelis-Menten equation, which is essentially S upon K dash M plus S. The just like an activator molecule, here also the Km gets modified to a new value 1 plus I upon Ki, and therefore, as the concentration of I increases, the value of Km also increases, and obviously the affinity decreases for the substrate. And the K dash M, essentially your modified Michaelis-Menten constant in the presence of an inhibitor, is your Km 1 plus I upon Ki. So, you notice here that the Km changes by in magnitude 1 plus I upon Ki, but the Vm remains unchanged, Vm into S on the numerator. There is no change on the magnitude of Vm. The maximum reaction velocity remains the same, but the Km changes. That is a very typical feature of identifying a competitive inhibitor. If you make a plot of a double reciprocal plot of or Lineweaver plot between 1 upon substrate concentration versus 1 upon reaction velocity at different inhibitor concentrations, you will notice that with increasing inhibitor concentration, the value of Km will undergo change, Km will tend to increase, or the slope of the profile will increase without affecting the point of intersection on y-axis, which is represented as 1 upon Vm. If you write the double reciprocal equation for the same thing what we wrote earlier, Km upon Vm 1 plus I over Ki into 1 upon S plus 1 upon Vm. So, basically as you notice that you get a same profile as I discussed, that the value of Km will keep on, if you plot 1 upon S 1 upon V naught, you get a series of profiles with increasing I. Essentially the slope increases, but the magnitude of 1 upon Vm remains unaltered. That is what we notice. Now, if you notice here also another fact, now one may be interested to determine the value of Ki in comparison to your Km and Vm, Km and the characteristic pattern of the inhibition, and you can use the double reciprocal plot as a basis for determining the value of Ki. Now, essentially if you look at the slope of the reciprocal plot, your slope is here Km over Vm 1 plus I over Ki, that is equal to Km over Vm into Ki into I plus Km over Vm. Now, essentially if you see, now if therefore, if you starting from your double reciprocal plot, you make a secondary plot which gives you a pattern of slope versus concentration of inhibitor. If you get a secondary plot from the primary double reciprocal plot, where you plot the value of slope obtained as a function of inhibitor concentration, you will again end up in a straight line with a slope of Km over Vm into Ki. Obviously, Km and Vm is the slope at I is equal to 0, which is already known to us, and therefore, the magnitude of Ki can be easily determined. Similarly, I mean you can also plot the apparent Km, either of course, the slope also has a meaning of the apparent Km. So, either you can plot apparent Km or you can plot slope, it will mean entirely approximately the same thing. Therefore, one can get the magnitude of Ki by plotting a secondary plot for these principles. So, to summarize the competitive inhibition, we must notice that the inhibitor molecule binds to a substrate molecule to an enzyme molecule at a site, either on the same site which is earmarked for substrate molecule or at a site in proximity to it, in such a way that interferes or restricts the binding of the substrate molecule onto the enzyme site, and therefore, the essentially it competes for the binding process on the enzyme molecule. There is no binding of the inhibitor molecule with the enzyme substrate molecule. Once the substrate molecule has bound to the enzyme, the inhibitor is ineffective. Either inhibitor has to bind to the enzyme or the substrate has to bind to the enzyme. Only that means the total enzyme concentration is distributed between inhibitor and substrate, and that is one. Second thing is the reaction, the enzyme catalyzed reaction in the presence of a competitive inhibitor essentially results in an increased Km value, and the Vm value is totally not effective, Third thing is that the effect of the competitive inhibitor can be successfully overcome to a significant extent if you increase the substrate concentration to a very large value as compared to inhibitor concentration, because they are competing on the same side. The concentration of substrate is much larger than inhibitor, then the possibility of substrate molecule binding to the enzyme is much more, and once substrate molecule is bind, the enzyme will no longer be available for inhibitor to act upon. So, these form basically, and finally the double reciprocal plot of 1 upon S versus 1 upon B at different inhibitor concentrations, followed by a secondary plot of slope versus inhibitor concentration, can be used to determine the magnitude of Km, Vm and Ki for a competitive inhibitor involved reaction. We will stop at this point.